Hi everyone. Today I'm going to talk to you about DNA affinity purification sequencing, or also known as DAP-seq. DAP-seq is a scalable transcription factor DNA interaction assay that employs affinity purified transcription factors to map transcription factor binding sites on naked genomic DNA in vitro. In this video, I'm going to discuss the differences between DAP-seq and ChIP-seq and also go over the DAP-seq protocol. So now let's discuss the differences between ChIP-seq and DAP-seq. As you can see, there are quite a few differences, but some of the important ones you should know is that ChIP-seq is an in vivo transcription factor binding site identification method, whereas DAP-seq is a high throughput in vitro transcription factor binding site identification method. ChIP-seq preserves native chromatin states via cross-linking, whereas DAP-seq utilizes naked DNA and recombinant proteins. ChIP-seq captures genomic sites bound by a given transcription factor, whereas DAP-seq is scalable to hundreds of transcription factors. How these methods are similar is that they both are used to identify transcription factor binding sites, and ChIP-seq data is often used to confirm the transcription factor binding sites identified through DAP-seq. Now let's discuss the DAP-seq protocol. First, the genomic DNA of interest is extracted. This can be accomplished using a phenylchloroform-based extraction protocol. Next, the genomic DNA is fragmented through a method such as ultrasonication, where you're left with genomic DNA around your target peak size of about 200 base pair. The DAP-seq library is then constructed by ligating adapters to the genomic DNA. Successful adapter ligation can be confirmed via gel electrophoresis. One thing to note is that genomic DNA used in DAP-seq retains chemical modifications such as 5-methylcytosines. An extra PCR step can be performed to generate a modification-free DNA library, see step 5. This method is known as AMP-DAP-seq, where the methyl groups are lost upon PCR and you're left with a modification-free DNA library. One quick tip. By comparing the two different library preparations, DAP-seq versus AMP-DAP-seq, you can study how DNA methylation affects transcription factor binding sensitivities. Transcription factor expression vectors are constructed and can be expressed in a plant or mammalian-based in vitro expression system. Next, affinity purification is performed. Transcription factors bound by affinity tags and cellular components are co-incubated with affinity tagged magnetic beads. Nonspecific binders are washed away, so you're left with only transcription factors bound to the magnetic beads. Next, the DNA library is then incubated with the transcription factors bound to the magnetic beads. Transcription factors bind to the genetic fragments in a sequence-specific manner. Unbound DNA is washed away, and the final product is then eluted. The eluted product is PCR amplified. An index sequence is added to each DNA fragment during the next-gen sequencing library preparation step, so that each read can be identified and sorted before the final data analysis. These samples are then processed through a next-gen sequencer, such as the Illumina HiSeq. The reads generated are analyzed, and the assembled reads are mapped back to the reference genome. Next, through bioinformatic analysis, transcription factor binding sites can be determined, and their corresponding consensus sequence can be defined. To wrap up this video, here are a few key points that you should remember. DAP-seq is a high throughput in vitro transcription factor binding site identification method. This method can be used to analyze how DNA methylation impacts transcription factor binding sensitivities. DAP-seq can be used to identify transcription factors that associate with DNA, and there's no need to create gene-specific antibodies or tag transgenic lines. DAP-seq is scalable, inexpensive, and a rapid alternative to ChIP-seq. Please refer to the references listed here for additional information pertaining to DAP-seq and the protocol discussed in this video. Thank you all for listening.